That's maybe 100, 200 amino acids versus thousands for most proteins. So it's still a very large molecule. And we know that peptides can have hydrophilic or hydrophobic qualities to it, but when we're using these for a hormone, they're mainly going to be charged. So that's going to make them hydrophilic. Then we can just take an individual amino acid, not 200, not 1,000. We'll just take one amino acid and we can use that as a hormone. So these are a lot smaller. We could take things like tryptophan. It has a ring structure, and then we can change it into serotonin. This is about all we're going to say about serotonin in this class. It's a really important neurotransmitter. If you take a neuro class, if you take any psych classes, you will hear about this a lot, but we don't talk about it in here. Tyrosine is another amino acid. You can convert that into catecholamines. Catecholamines are just dopamine, epinephrine, norepinephrine. We're going to talk about those a lot because those have a lot of effects throughout organ systems. The other thing we can change tyrosine into is a thyroid hormone, which we'll also talk about in depth, just not here. We have a class of proteins called steroid hormones, and they're coming from cholesterol. So you take a molecule of cholesterol, it's fat, and you make all kinds of changes to them, and you can come up with this huge range of hormones. These are really important. We're going to have sex hormones. We'll talk about those mostly when we get to the reproductive system. We're going to have things like cortisol and corticosterone. Those are really important for functions like stress response, things like that. Aldosterone, we're not going to talk about till we get to kidneys. And then calciferol, I'm going to talk about briefly in a story that we start with, but this is what we call vitamin D, which is odd because it's not a vitamin, but that's what we call it. Okay, and vitamin D is really important in being able to absorb calcium. So we can have calcium that we've ingested, but we won't actually be able to absorb it and use it unless we also have vitamin D to help with that. So we will go through these hormone types and talk about which ones are hydrophobic and hydrophilic, and then because of that, what receptors they have to use. But before we get to that, two things. There was a skin story I wanted to tell you yesterday, and I ran out of time, and so initially I was going to drop it. But given recent events, it's become something that I really want to tell you again. This is going to be, it's a Walren story, okay? So I don't know this in the same detail that he does. And I'm going to give you an abridged version because we don't have a whole lot of time. But this is the melanocyte story. So this starts way back in the beginning of man. By the way, don't worry about history. None of that is testable. Do understand melanocytes, which will be like 2 seconds of the story. But way, way back when man first started, when man broke off from the other primates into Homo sapiens, this was back in Central Africa in the rainforest. Okay, so they initially had fur. They couldn't walk upright. But as they continued to evolve, they began to walk upright. And they were able to move out of the rainforest and start hunting on plains of Central Africa. Now, Central Africa is on the equator, so you've got sun that just comes down on you constantly, and you've got these early humans that are covered in fur, and they're running around chasing after other animals to eat. It got really hot, and it wasn't good, and so there was pressure to lose that fur. Over time they did, and now we're, depending on who we are, we might be fuzzy, we have some hair, but we don't have a layer of fur. So that was great for them in terms of being able to handle the heat, but now it's a problem because they've got sun rays coming directly down on them, onto their epidermis. Sun has UV radiation. It carries photons of UV radiation. One of the most damaging is UVB. Okay, UVB is really good at breaking ring structures. So we know this now. We know that we have to avoid UV in too high of a quantity because it can cause damage to DNA, because DNA has a lot of ring structures. They weren't terribly worried about DNA. Cancer is one of those things where you do the damage now, you see the results decades later. They weren't typically surviving that long. But that's not the only thing this can break. This is going to break any kind of ring structure, and we know a lot of our amino acids have that ring structure. So we're going to be destroying a lot of proteins here. There began to have a selection pressure to try to do something to protect against this UV light that they were facing every day. So we told you about the keratinocytes, and then I added in stem cells on top of it. Now I'm going to tell you There's another cell in here. In the stratum basale, called the melanocyte. Melanocytes produce melanin. Melanin is a pigment. Not unlike henna, if you release this pigment, the cells can take it up. So you will get all this pigment in your keratinocytes. And just like henna, you can see it. So if you have more pigment, you're going to have a darker skin tone when you look at your skin. The great thing about this melanin is that it actually acts as a shield against this UV light. So you could think of it as sunscreen for the cell. Over time, they developed more and more of the sunscreen. They were up at like SPF 50. Really nice dark skin tone in the process. Everything was great until people started migrating. And when they migrated north and started splitting off to head into East Asia and into Northern Europe, they ended up in latitudes where the sun is now no longer direct. So these UV photons are coming in at an angle. In addition, it's cold, and they've lost their fur. So now they have to go steal fur from other animals and wear it. There's advantages to this because now they can take it off when it gets hot again. But this is going to block a lot of sunlight from their skin. And you might think, great, UV bad, they should be in good shape. Except that there's one molecule we really need, that we do need the UV for. So we said that cholesterol can be made into all these different kinds of hormones, including calciferol, or vitamin D, which is super important for being able to absorb calcium. And we know calcium's important. You don't know why yet. We'll talk about calcium when we get to each unit, how it's used. But we know it's important enough that we will actually break our own bone to get it out and put it back in our blood. We can make this cholesterol by breaking a ring in the cholesterol. So now these individuals who had moved out into East Asia and to Northern Europe were not getting enough cholesterol broken down into the calciferol. You can get some vitamin D from your diet, but we still need to make some ourselves as well. So now they had to go through another selection pressure where they had to reverse their previous mutations. They didn't actually directly reverse those mutations, they were new mutations, but they had to start decreasing the amount of melanin, the amount of pigment. These two different populations that went to East Asia and Northern Europe, they had completely different mutations, but the same effect. Less pigment, lighter skin tone, more UV coming through to make vitamin D. And again, everything's good. Probably not, it was a hard time for people back then, but in terms of making vitamin D, things were good. Until maybe 10,000 years ago, when we had another major shift, we started moving from hunting and gathering into agriculture. So now we're using more plants, we're eating grains, and we're not getting as much vitamin D in our diet. We need to go through another round of reducing this melanin pigmentation so that we can make more calciferol through the UV. And so all of these different changes that we've had, based primarily on where people have moved back and forth from, changes in diet, have now led to this incredibly diverse array of skin tone just based on the amount of pigment that you carry in epidermis. I think this is one of the most amazing examples of an evolutionary pressure because it's not only something you can actually see, you can visually note, but it's also an example of where we had to push it one way, and then when something changed, we had to push it back in the other direction. Unfortunately, this story is not all rainbows and puppy dog tails, because as we all know, somewhere along the line, people started thinking that they could use this level of pigment to actually make judgments or measure people's character or their intelligence or their worth as human beings. And that's just bollocks. It's pigment. Having more or less pigment has absolutely no effect on an individual's worth as a human being. So that's I'll get off my soapbox, that's my great example of an evolutionary pressure and how it affected our physiology, but we need to move on now to osteoclasts. Yes?
I just have one question. So, if you're out in the sun for really long and like you can.
Yes.
What is that about? Is that your body producing more melanin?
Exactly. So if you have enough melanocytes to produce melanin, when you're out in the sun and you get that UV exposure, it will trigger it to pump out more of the melanin to protect from the UV. It's like, oh, you know, there's a lot of UV here, there's damage, let's put up more shield. If you're like me, you don't have a whole lot of melanocytes, you just don't produce a whole lot of melanin. I go outside, I turn into a tomato. Eventually I get more freckles, but that's pretty much the extent of it. Yeah. Okay. So, bones, we got through all the bone except for osteoclast, and we said that an osteoclast is going to be a bone breaker. And we know that we can do this for several reasons. We may need to just break this bone down to pull calcium out because we're low. We may need to do this to remodel our bones for some reason. Okay. We can actually do this as well when we have damage. Maybe we need to clean up some of the damage before we can fill it back in, so we want to break that down. If we were to look at just the shaft of a bone is what I'm going to show. Okay, so imagine that this is, well, we'll say, well, it doesn't really matter. We'll say it's the radius. So it's just a long bone here we're looking at. We're going to have these osteoclasts. And these osteoclasts will carry hydrogen ions. Remember that this bone, this long portion of the bone, is compact. It is made of osteocytes arranged in osteons, and then it has the collagen fibers, and then that cement, that hydroxyapatite that is holding it all together. It's calcified this entire bone. So, hydroxyapatite is calcium phosphate. Among other things. What the osteoclast is going to do is attack this calcium phosphate. It will dissolve the cement that's holding everything together here and allow it to break this bone portion down. So when you put this hydrogen ion, this acid, right onto this calcium phosphate, you can break it into calcium ions and phosphate. So if you're just breaking it down to get to your calcium, no problem. You free the calcium, you stick it in your blood, and you can use it. Okay, if you're actually remodeling, you might break this section down, and then maybe you're building up somewhere else. So you'd have osteocytes in here. Osteocytes that will lay down more extracellular matrix and make more bone. Okay, so osteoclasts will make the bone, osteocytes will make the bone, osteoclasts will break the bone. Questions on bone? Yeah.
Is this process going to be happening at the same time? So like if more osteocytes are making bone, osteoclasts breaking down the bone, or is it like going to happen at separate times for different reasons?
So do the osteoclasts and the osteocytes work at the same time? It depends on what it's doing. If the osteoclast is just breaking it down to scavenge the calcium, it's probably working on its own. You can't build more until you get more calcium in your system. If you're rebuilding, you're probably breaking down and rebuilding new at the same time. Yes?
Why did you say we want to bone break? Is this for the growth period? Like where at that time of our lives where we need to elongate it? Or is this for...
Great question. So are we doing this like in the growth plate while we're trying to grow? Yes, to an extent. While you're growing, you have to actually make sure that you're growing it in the proper directions and that it's formed correctly. So you may have to break and rebuild a little bit there.
What's the main point of this? I've missed that.
The main point of this is just that this is how we can break our bones down when necessary. So, you know, just any kind of rebuilding that we might need throughout our life, or if we need that calcium.
Can this osteoclast go anywhere on the bone, or is it limited to specific areas on the bone?
Can osteoclast go anywhere on the bone? Yes, but it's a lot more complex in other areas that aren't compact bone. So I'm just doing the long bone, compact bone example. All right. Yes.
According with the process, like the height drops, like height is again, is that the process the osteoclast is doing to break the bone?
So the osteoclast will put this hydrogen ion directly onto the bone surface, and there's hydroxyapatite there. So when you expose this calcium phosphate in the hydroxyapatite to a hydrogen ion, to that acid, it will break it down. So it's basically uncalcifying that matrix. Okay, we'll move into endocrine. So again, endocrine is intercellular communication by using these chemical signals called hormones that will go to different parts of the body and bind to specific receptors to cause a reaction. We're going to talk about the receptors and the different reactions, responses that they can have on Monday. But just to introduce you to the system itself and the receptors. Intercellular communication. This is going to require one cell that sends a signal. So you have a sending cell that releases hormone. And then you have a receiving cell that will bind that hormone and respond. So our signaling types. We can have autocrine. That means that this cell will exocytose this hormone and a receptor on that very same cell will bind it. Okay. Self-signaling. We can also do paracrine signaling. In paracrine signaling, you're going to have your sending cell that will exocytose the hormone, and the neighboring cells can bind it. So as this diffuses away, it'll bind and cause a response in the neighboring cells. Just like oxygen, most of our molecules have a diffusion limit. They can only go so far before they get degraded, or they're in such low concentrations they've diluted to the point where they can't signal anything. So if we need to go further than just our neighbor cells, we can use endocrine. We'll have a sending cell. It's going to release hormone. But in this case, a nearby blood vessel can pick this up. Remember, there's always going to be a blood vessel nearby. All cells have to have oxygen from the blood in order to survive. So this can now diffuse into this blood vessel. I'm going to draw lots of knots. And then, in a galaxy far, far away, actually, that's not really fair to say. We could be sending it someplace that's not like the opposite end of our body. It's just further than diffusion limits. So it's not something that could diffuse. It has to go through blood. But it could be going from, say, the adrenal gland, which is directly on top of your kidney, down to the kidney. Too far to diffuse, but still not that far away. So here is your receiving. It's going to have the receptor. There'll be another blood vessel that's close. And some of this hormone will be in the blood. This hormone can diffuse out and bind and cause a signal. Notice the difference here where I showed this sending cell releasing a lot of hormone, but the receiving cell only seeing a little bit of it. And hopefully that makes sense. I think of it as like having a 500-gallon aquarium full of water, and you take food dye, and you pour the entire bottle of food dye into one corner. That's a lot of molecule that you put into that one area. Come back an hour later when it's diffused through the whole system, and then take a sample of that water and see how many molecules are in each little area of water. And it's going to be, maybe you put 1,000 molecules in with the dye, but now you're looking at it and there's only one per area of water. When you diffuse it, the concentration goes down. And this is important because it means that our hormones are not going to be in high concentration. So in order for our hormones to actually function, we have to have receptors that are okay with working with these low concentrations. So the typical concentration of a hormone is going to be in the range of 10 to the negative 9 to 10 to the negative 12. Those are really, 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 really small numbers, lots of zeros. So that leads us into receptor characteristics. Any questions on signaling yet?
What would be an example of an autocrine, paracrine, and endocrine cell? Or signal?
The only autocrine example I can think of is actually like a negative feedback, where you're releasing hormones, and then when you have a high enough concentration, it will actually be able to bind here and signal to the cell, you've released enough now. I know it's used for other things, but I don't have examples of it. Paracrine is, we'll see this in the digestive system, where we can release a hormone from one to signal others that they need to release acid into the stomach for digestion. And then endocrine is what we're going to talk about most. Most of our hormones are actually endocrine hormones. So pretty much everything we've talked about there is going to be endocrine hormones going through the blood. So released from things like adrenal gland or brain, and then going to all areas of the body.
So then paracrine is by diffusion and endocrine is just too far for diffusion limit?
Paracrine is diffusion to its neighbors, and endocrine is too far for diffusion. It has to be brought there by a highway, by blood vessel. You had a question, Allie?
What was I saying? Like after the hormone binds to the receptor or whatnot, what happens? What is the stuff?
So the whole signaling sequence when a hormone binds to these cells is something that we'll get into in Monday. There can be several different responses, and we'll walk through what those responses are.
So how does it pick which one it's going to do? Is it just there's only receptors for autocrine release hormones that on that same cell? Or does it just release a small enough amount that it can get to a blood vessel? Or how does it pick which one it's going to do?
how does it pick which one it's going to do? It really doesn't. It will release out, and if there is a receptor right there, then it will bind to it, and that's an autocrine. If there's no receptor there, it'll diffuse away. If there happens to be receptors on neighboring cells, then it binds there, and we call that paracrine. If there's nothing there, or maybe there is, but it still gets into the blood and it happens to find receptors elsewhere too, then we say that's endocrine. So it's not that any one hormone is any given system. It's just that when we bind to the same cell versus neighboring versus it goes elsewhere, we call it autocrine, paracrine, endocrine. Okay? All right. Let's talk about the receptor. So we want receptors that are very specific. We want a receptor that is only going to bind to a particular hormone. We don't want receptors on our cells just responding to every hormone that we put out. That would be very confusing. But some hormones have varying levels of specificity. So you can have a receptor that looks absolutely nothing like that, because these are 3D structures and way more complex than this. But for purposes of an example here, and I'm going to draw that a little bigger. So that could be one receptor. You can imagine if you had 3 different hormones. You had a hormone 1. You have hormone 2. And hormone 3. Now, just looking at this, hopefully you all recognize that hormone 1 is the one that should bind this. Question?
You mean by receptor characteristics, right, not hormone?
I'm sorry?
You mean, you wrote hormone characteristics, but I thought you were saying.
Yes, thank you for catching that. We're not talking about the hormones themselves here. We're talking about the receptor characteristics. Thanks for that. Okay, so if hormone 1 is meant to bind in there, no problem, it's going to get in there. I shouldn't say no problem, but hormone 1 can fit in there and bind nicely. Keep in mind that these things are just going to be randomly bumping around. You release the hormone, it diffuses around. It may or may not line up correctly to actually get into its appropriate hormone receptor. Okay, so if this actually happened to come in this way, it wouldn't be able to fit into that binding pocket. It has to line up. That's one of the reasons that we don't have a 1-to-1 ratio. We have to have a higher concentration of hormones to actually be able to bind these receptors, because not all of them are going to be able to fit in correctly. But maybe this hormone too, if it lined up just right, could also fit in there. It depends on the receptor. If the receptor is highly specific for its hormone, it will not fit anything else. But sometimes you have receptors and hormones that are close enough. Some of these hormones came from the same thing. We said we took cholesterol and then we made it into, all these different steroid hormones, basically just by changing one thing here or there to make each new one. Some of them may be close enough in size that they could fit this receptor. If it has a high specificity, it's very specific for its hormone. If it has a low specificity, it may be able to fit similar hormones. We'll see this a little bit with things like epinephrine and norepinephrine, because they are so close. You can have an epinephrine receptor that may occasionally bind norepinephrine as well. It won't bind it as well. It's not quite the right shape. And so it will not have as high of an affinity. That's the second factor. Affinity or stickiness. This hormone can come in, it can bind, but it will not stay there. It will eventually dissociate and release. How well it's held and how long it's held is a matter of affinity, or how sticky that receptor is for that particular hormone. So in this case, we could say with this example up here that hormone 1 had a high affinity. Hormone 2 could get in there and it could bind, but it's not going to bind very well because it's not quite the right shape. So there's a lower affinity for hormone 2. Okay. We can actually quantify affinity, not just say, hey, it's sticky. We can measure this, although we won't make you do the calculations, but we can measure this as KD, or dissociation constant. The definition of a dissociation constant is the concentration of hormone required to occupy 50% of the receptors. Okay, that might sound a little bit like gibberish, so let me just show you how that would work. You have your cells here, and they had that receptor on it. Okay, two things. You would never just have 4 receptors for something. I can't imagine a single hormone that would only have 4 receptors. Also, we know that hormone concentration is typically really small, but I don't do well with small numbers. I don't do well with big numbers. I can't wrap my head around that many zeros. So I'm going to use an example where I'm using even numbers. Just remember that hormones are never in this kind of concentration. So if this example here, we're binding hormone 1, in order to get 50%, which would be 2 of these, to bind on average, and these are going to bind, they're going to dissociate more, going to come in, they're going to bind. So on average, if 2 of your receptors, 50% of your total receptors have been bound by your hormone, we could say, we'll just make this up, we'll say that the hormone concentration to reach this state is 1 molar. So if you put 1 molar of that particular hormone in, you will bind on average 50% of your receptors. If you increase your hormone concentration, you'll end up binding more, 60%, 70%. If you put in less concentration, you bind fewer. Okay? This is your KD. So now you could say that the KD here equals 1 molar. If we did this experiment again, and we took these same cells with the same receptors, but now we're testing out hormone 2, this is not going to have as high of an affinity. This receptor is not meant for this particular hormone. It might be able to bind it, but it will not bind it as well. It will not be as sticky to it. It can't hold on to it. So in this case, we might find that we would need a hormone concentration of 2 molar in order to bind 50% of our receptors. That means our KD is 2 molar. Okay. Notice that we said this one had a higher binding affinity. And you could see that our KD is smaller, it's 1 molar. This one has a lower affinity, and the KD is higher. It's 2 molar instead of 1. This is always the case, okay? If you have a higher affinity, because you can bind it better, it's stickier together, then you do not need as much concentration to be able to fill those receptors. So we have a lower KD. I would really love for you guys to wrap your head around why that is. But if this were the only thing you walked away from today, I might cry myself to sleep, but you'll do just fine answering these questions on the exam. Okay, so this is your takeaway. Yes?
So when hormone 3 has 0 affinity?
Hormone 3, perfect. Hormone 3 doesn't fit at all. Okay, so there's no specificity for it. It wouldn't bind. Your KD would be, I don't know, 100,000, because like infinity, really. No amount of concentration would ever get it to 50%.
So then does that mean that like a ascending cell would just have to release like a lower concentration of hormone in order for like a receptor with higher affinity, in order for enough?
Yeah.
Does that kind of make sense?
I think I get your question. So let me say you've got this cell and you've got this receptor. Let's just say that this was epinephrine and this hormone was norepinephrine. This is an epinephrine receptor. It's meant to bind epinephrine. So you can release epinephrine in those quantities and you'll be able to bind just fine. This can also bind norepinephrine a little bit, but you would need higher quantities than normal to really be able to get this to bind and function. Okay, so if there's a lower specificity and something else can bind, you would need a higher quantity than normal. You might need 10 to the negative 5th hormone concentration to get binding with not quite the right hormone.
So if like a receptor has a lower affinity, then it's going to need a higher concentration of hormone in order for a higher amount of hormone to bind.
Yes, lower affinity is going to require higher concentrations. Okay, how are we doing? Oh dear, we have 9 minutes. All right, let's talk about hormone types. So we're going to talk about the hormone. We're going to talk about what it's made from. We're going to say whether or not it's hydrophilic or phobic. And then based on that, what kind of receptor it can bind. So our first one that we mentioned was peptide or protein hormones. We know that peptides and proteins are made from amino acids. Typically, amino acids can be either hydrophobic or hydrophilic, but the ones that we use for hormones will usually be hydrophilic. Because it's hydrophilic, it has no problem being in the cytoplasm, it has no problem being in extracellular space, but it cannot move through the membrane. The membrane is hydrophobic in the center, and a hydrophilic molecule is not going to like trying to do that. So we're going to use transmembrane receptors, which is basically what I've been showing this whole time. We can also have our modified amino acids. So we can have serotonin that comes from tyrosine, tryptophan. Tryptophan is hydrophilic. When we change it into serotonin, it stays hydrophilic. And therefore, we'll use a transmembrane protein. We can have our catecholamines, which is our dopamine. Oh, let's change color. Dopamine, epinephrine, and norepinephrine. All of those are coming from tyrosine. Tyrosine is hydrophilic. All of those will be hydrophilic. And therefore transmembrane. We can also make thyroid hormone from tyrosine. But in this case, even though tyrosine is hydrophilic, when you change it into thyroid hormone, you make it hydrophobic. Hydrophobic cannot bind to transmembrane proteins. It doesn't like being out there in the extracellular space in a hydrophilic environment. These are going to bind instead to cytoplasmic receptors. Okay, and then we have our steroid hormones. Those include our sex hormones, our aldosterone, vitamin D, they're all coming from cholesterol. Cholesterol is a fat, so these are all hydrophobic. All steroid hormones are hydrophobic and will bind to cytoplasmic receptors. I know there's a lot of steroid hormones listed here. We're not going to expect you to memorize them all. You can typically recognize a steroid hormone. Some of them are really nice. They just have sterone in their name. So that's steroid hormone. It's nice when they do that. The rest of them kind of tend to have these alls: cortisol, calciferol, estradiol. Okay, so if you see something like that, you can pretty much bet it's a steroid hormone. But we're not just going to give you some random hormone and expect you to recognize it. We'll give you clues in the passage as to what kind of hormone it is. Okay, we actually have time for questions.
Are peptide, protein, serotonin, and catecholamine all modified amino acids?
So peptide proteins, we wouldn't really consider a modified amino acid. We're just creating a protein from a chain of amino acids, whereas these, you're taking one amino acid, changing the structure of it to create a new molecule. Yes?
How would you know if they were hydrophobic or hydrophilic?
So you should know that your steroid hormones and your thyroid hormone are hydrophobic. Everything else we've talked about, protein hormones, modified amino acids except thyroid, will always be hydrophilic. Man, I just rushed through that. I didn't think we'd have enough time, but you're actually getting out early, so happy weekend. Thank you, guys.